Well, hi, everyone. Um, I am Karen Pierce, and I'm really excited to be here today to talk with you about SPARK, the largest genetic study in the nation. I am a professor in the Department of Neurosciences at the University of California, San Diego, and the co-director of the Autism Center of Excellence. And SPARK is an amazing study because it is the largest study of autism. The goal is to recruit over 50,000 families so that we can figure out what makes us unique and how we can all lead better lives. I want to first introduce my co-host, though, Michaela Getz, and she's running the entire show, and she helped put on this webinar, and she's also going to answer any questions that you guys might have along the way. So SPARK stands for Simon's Powering Autism Research for Knowledge. And the goal of SPARK is we want to speed up research and advance our understanding of autism so that we can help improve lives, because every individual deserves to reach their full potential. Now, autism is strongly genetic, but also has an environmental impact. We know this because identical twins share 100% of their DNA. And if autism was 100% genetic, both twins would have autism. But the reality is that 77% of twins have a concordance. There's a 77% autism concordance rate for identical twins. Now, fraternal twins who share 50% of their DNA, but they share a womb, their rate of autism is actually 31%. And if you compare that to individuals who are just siblings, they're not fraternal twins, they're usually a year or two or more apart, they share the same amount of DNA as fraternal twins, but the concordance rate for autism is 20%. So you can see that the difference between 31% and 20% is that because these fraternal twins, they share a womb, they're exactly the same age, their environment's exactly the same, they have a higher concordance rate. And then scientists say, okay, the environment must be having a little bit of a contribution because it's a higher rate than just siblings. Now, let's just, go back a little bit. I'm going to do a tiny bit of genetics review, and not a lot. We basically know that there are around 20,000 genes in the human genome, and every single cell in our body contains all 20,000 of these genes. And genes are contained on chromosomes, and we have 46 chromosomes. 23 you got from mom, 23 you got from dad. And our chromosomes contain the DNA. And the DNA is essentially just a long sequence of genes. And basically, our genes are made up of nucleotides. These are adenine, thymine, guanine, and cytosine. And if there's even like the tiniest change in one of those letters, it changes the gene meaning. So here on the slide, you can see it's just a partial sequence of a gene. We have ATG, TAA, GCC, etc. And if one of those letters gets changed accidentally, it can change what the gene is supposed to be doing. So basically, it changes kind of, in more technical terms, what protein it's supposed to be coding, or how the protein is acting. And if you think about just a regular English sentence, we have Bob the cat cannot sit. And if you change even one letter, like you can change the C to a B, it changes the meaning entirely. And so it's the same thing with our genes. You can change a single letter and it completely changes what the gene is supposed to be doing. And so when we think about these changes, there are different terminology for it. So most people call these de novo genetic changes. And a de novo genetic change just means a new change. And it's a change that occurs either in the mom's egg or in a sperm cell or during fertilization. One of those letters gets changed, maybe during cell replication. Lots of things are happening when all of these cells are being made, and that can cause a little bit of an error. And it's important to note, though, that these de novo changes are present in everyone, and they can occur anywhere in our genome. So every person watching this webinar right now has some sort of a de novo change. You've got a single letter that has been changed, but it's not having an impact on you. But certain genes, when they have that change, can be related to autism. And scientists believe that genetic factors, they're de novo, but sometimes they can be inherited, meaning that there is a genetic change in mom and dad that's passed down to the child. And scientists believe that autism is caused, probably around 80% of autism is caused by genetics, maybe even as high as 90%, some scientists would say. So the environmental factors related to autism are pretty tiny, but that's around 20 to 10%. And it could be related to pollution from cars. Mom was ill during pregnancy, advanced maternal age, so if you are an older when you conceived, that increases risk of autism, or taking some sort of medicine like valproic acid while you're pregnant. And so the question is, how is Spark going to use this genetic information to speed up autism research and discovery? So we're going to recruit at least 50,000 families who have a child with autism, and we're going to collect some phenotypic information because we want to understand what are the genes doing in relation to someone's personality traits or someone's cognitive or linguistic abilities, and then collect also the genetic info. And then we're going to return the genetic results to the families so that individuals who have the same genetic issue can be grouped together, and that gives us more power to learn things. And then we can match participants to additional research studies if they're interested. And then this whole thing is going to bring us towards precision medicine, because right now, you're a parent, you receive a diagnosis of autism. There's not really a lot out there that lets you know which treatment might be optimal for your child, kind of what biological issues are going on. And so that's why we need to get this genetic information so we can kind of move towards precision medicine. Right now, we've got over 30 research partners collecting saliva samples from families across the country. UCSD and also SARC, the Southwest Autism Research and Resource Center, headed by Chris Smith, is another one. And together, we have collected saliva samples from over 100,000 individuals with autism and 175,000 family members, making this an unbelievably large study. But we always need more people, which is why you're on this webinar. So I'm hoping you'll join. You'll join the mission because Spark is amazing. And we are returning genetic results to around 10% of families. So that means when you provide your saliva sample, at 1 out of every 10 people will have a positive genetic finding, and we will mail you a report. And I'll get into that a little bit more in a minute. And these genetic analyses have increased the number of genes that we know are associated with autism. And Spark actually does a really wonderful thing. They put out this thing called the Spark Gene List, which essentially provides the public all of the genes that are relevant for autism. And when we first started in 2017, there were only 78 genes on the Spark Gene List and 11 copy number variations. The most recent gene list has been published in 2022, and we have 162 single genes plus 43 copy number variations. And what's great is that, like I said, they're publicly available, and we'll mail you guys a PDF of this entire presentation, so you don't have to worry too much about taking notes. But the gene list is published here at this website. And then they also have what's called genenames.org. It's actually not supported by Simons. It's an independent organization that you can type in any of these genes, like P10, if let's say your child has a positive finding, or you want to just learn more about particular genes, and it will tell you a lot more about genes, about particular genes that you've designated. And this is really important because when you go on the internet, sometimes it's not clear, if this information is credible or if it's the best information. But the Simons Foundation really curates the information with a lot of rigor, and really the best scientists are involved in this. And so this information is really top notch. And the way you participate is you just basically spit into a tube. So when you sign up, you'll get a box in the mail. If you've ever done 23andMe or anything like that, it's very similar. You spit into the funnel, then you twist the lid off, and then you screw the cap on. And so you're basically spitting into a tube and you're done. But obviously, some children with autism. So the goal is we want mom and dad and child to participate. There are situations where sometimes maybe you're divorced, or the biological dad is away on deployment or not living anymore. We want everyone to participate. So if you're a single mom, participate, or if you're a single dad, it doesn't matter. The goal though is to have all 3 people, mom, dad, and child. But sometimes, that's just not, that's not going to happen. And I see that there have been a couple of people raising their hands. I will answer questions at the end. So we've got plenty of time. So some kids can't spit in a tube, understandably. So we've got this assisted saliva collection method where it's essentially a sponge, and you just sort of put it in your child's mouth and depress against the child's cheek. And we have people who can come and help you. Michaela, as I said, is the study coordinator. She can come to your house. So you don't necessarily have to do this alone, but it's pretty simple. And then you just take the little sponge and depress it against the plastic so the saliva falls out and fills the tube. And so this is what's called the assisted saliva collection method. And then you put your saliva in a box, mail it back to us, and then we send it off for DNA sequencing. And you probably are wondering, like, it seems a little mysterious, what kind of sequencing is done. And basically here at the Simons Foundation Project, we do what's called whole exome sequencing. And whole exome sequencing is a genomic technique for essentially encoding for sequencing all of the protein coding regions of the genome, which is called the exome. So basically, here's just like a little cartoon of your DNA. You can see there's a lot of stuff in there, and the exon is really only a tiny part. So I made a little graphic here with this apple. The exome is only about 1% of the whole genome, but it's a really important part of the genome because it's the part that encodes for proteins, which provides instructions for things like your hair color, your eye color, your cognitive ability, also your brain development. So that's really where a lot of the action is happening. And so that's why this Spark study is doing whole exome sequencing. And then if you want to sign up, which we hope that you will, here's a little bit of a close-up of how you can participate. So you basically register online at sparkforautism.org. And it's important that you sign up with the /UCSD.SARC. So UCSD is us, University of California, San Diego, and SARC are our partners in Phoenix. Because otherwise, if you just sign up at sparkforautism.org, it just goes into the larger big pool, and you're not associated with our site here in San Diego and Phoenix. So it's important that you designate that you are a sparkler from San Diego or Phoenix. And then they'll ask you to sign some consent forms to share your data, that you might enter some basic information about your family structure, like how many people are in your family. Then you have a separate consent for your DNA analyses. You'll complete a couple of surveys, like asking where did your child get their evaluation. Then after you've filled out all of your consents, we'll mail you the box and you provide your saliva sample. And then the DNA sequencing happens. And then only around 10% of families will get a result, but you will, around 10% of families will get a result. It takes a while because it's research. And as you saw, we have over 100,000 individuals with autism who provided saliva samples. It takes a really long time to get all those processed. So it could take up to a year, but it could be as fast as 6 months. But the nice thing is, once you provide your saliva sample, once we discover new genes associated with autism, we will reprocess your sample about once a year. So let's say you don't get a result. We only will notify you if you have a positive finding. Let's say you don't hear anything. You're like, okay, I don't have anything. But maybe in 2 years later, you might, because we reanalyze it about once a year. And then once you get a result, let's say it's a P10 issue, we will invite you to participate in Simon Searchlight, which essentially is a big network of families that all have the same genetic issue. So there'll be, a bunch of families that have a P10 issue, another bunch of families might have a SHANK2, another bunch of families might have another gene. And so you're with a community of people who have a child who have all the same genetic issues that your child has. So it's really nice. And it also helps speed up research when we can push everybody together who have the same genetic issue. And here's kind of what it looks like when you go onto the website to sign up. We'll say, oh, please tell us about your child. Then there's the initial consent form. Then there's the diagnosis information that you'll fill out. And it takes usually around 20 minutes to sign up online for this study or for this. It's really not even a study. It's a project. It's a big family. And I'll talk with you about other benefits of joining, like, webinars that are super interesting and some money that you get. And, just a lot of trusted information is really provided with this particular project. And just so you know, I'm an autism researcher. I do a lot of different types of things like brain imaging, eye tracking. And this study, though, I am just one of literally over 100 people involved. And I do it out of a sense more of altruism and wanting to support something so important. I am not going to be a first author on a paper. This is not really my research project. I am just like a soldier supporting it because I believe in it. And as an academic, you have to write your own grants and your own papers. Otherwise, you won't survive. But I take the time out to participate in this project because I think it's that important. And that's unusual because, it's very unusual to do something like that. So that just shows like how important I think this project is. So once you sign up, you'll get your dashboard, and the dashboard will say, oh, please complete some surveys, basic information about your child. Then it tells you your saliva kit status. It tells you where your kit is. Is it on the way to the lab? Where is it? And then if you want to sign up for any other studies, your invitations are here. And the important thing to know about this is we will never call you on the phone or go to your house to sign up for studies. All that will happen is we'll send you some emails saying, hey, there's this new study opportunity. Are you interested? And it's mainly just online surveys because people from this project are across the entire United States. And so a lot of these studies are just online surveys, and you probably will get a gift card and you'll be helping out with research if you do additional surveys beyond just the surveys associated with your saliva sample. And the benefits of joining is you will receive a $50 Amazon gift card. You'll get access to monthly webinars, newsletters, and dozens of really accurate and informative articles. You get the opportunity to participate in other studies. You get the knowledge of genetic results of your child. Again, up to 10% of people will get a result. Also, the important thing too is you will get complementary genetic findings. So if we do find something, we're not going to just say, hey, you have this genetic finding. The Simons Foundation will pay for free for you to talk with a genetic counselor who has expertise in the particular issue, the gene issue that your child has. You'll join a community and feel more connected with other families affected by autism. And you'll contribute to historical research. This is historical to have a study this large. This is the largest study of autism in the world, as far as I know it. And you might be thinking, well, how is my, information going to be protected? This is super important, and Spark goes through a lot of lengths to make sure it is protected. First, all data are encrypted at all times in a secure database, and data are stripped of personally identifying information whenever, because the whole idea is we want to share information with all other researchers. Maybe somebody has a great idea about genetics, but when they get your child's genetic information, there's never any name or anything associated with it. Just a little barcode or a little number letter identifier. So nobody knows the names or the addresses or the birth dates of anybody in this particular sample. They can only access the data if they agree to protect the data and use it only for scientifically valid purposes. But most importantly, Spark holds a certificate of confidentiality from the National Institute of Health that prohibits access to the data by non-researchers without the participants' consent. So basically, researchers can use the certificate to disclose information that may identify you in a federal, state, or civil or criminal lawsuit. For example, if there's a court subpoena, that means researchers can use a certificate. Let's say something happens to you and you have to go to court for something, and they try to get your child's DNA. The researchers will be like, no, you're not allowed to get this because we have this certificate. We can legally refuse anybody access to this because this is a federal certificate. And Spark will inform participants of other research studies, but they control what they want to participate in. And so we've learned a lot of things. We've identified new genes through the Spark project. We know that remember I was talking about these de novo changes or de novo mutations that count for around 20 to 30% of autism genetic risk. And continued research suggests that there's still hundreds of genes that are linked to autism, probably more in a background genetic sense, but we need to continue to sequence as many families as possible so we can understand this process. And here's just an example of a paper that was published by Pamela Feliciano and colleagues at the Spark study site. And here we can see that basically this research paper identified a brand new gene associated with autism called BRSK2. And BRSK2 is an interesting gene because it's a regulatory gene, and that means it's a gene that regulates what other genes are doing. It affects a lot of processes such as cell division and axonogenesis. So this is kind of a critical gene to learn about autism, and they found it because we have so many families, participating. They had the large sample sizes that allowed them to hone in on this and make this discovery. And if you are a family that has a positive finding, here's what's going to happen to you. You will get around between 6 to 12 months of your trio completion or your duo completion if you're only just a single mom or a single dad. Then you'll get something mailed to you, and you can review this with your own medical provider, or as well, again, you can get a genetic counselor that is paid for free by Spark, and it will look something like this. This is what the report's going to kind of look like. And then we'll also mail you a brochure about your genetic finding that gives you some more details and just, gives you a little bit more information. The other thing to know about this project is, I don't know if anyone of you guys has ever done 23andMe, but there are a lot of genes that are associated with other medical conditions that are not related to autism. And you have the right, if you want it, we will sequence, when we do your sequencing, we'll check for 3 non-autism related conditions in you and your family from the Centers for Disease Control Tier 1 list. So we can check for breast cancer and ovarian cancer syndrome. So HBOC is a hereditary gene, meaning that it is passed down in families. And this is, if you have this, then you will have greater risk for breast cancer. So you can, when you're signing up in your consents, you can say, yes, I want to find out about the breast cancer gene. You can also say, if you want to find out about Lynch syndrome, which is a hereditary disorder, associated with an increased predisposition to colorectal, endometrial, ovarian, and other cancers. So it's really important to know if you have this. Then there's also familiar hypercholesterolemia. Let's just call it FH. Basically, some people have really high cholesterol. It's because they've got the gene for it. And this study will screen this for you. And if you say, yeah, I want to know, they will let you know. If you can say, no, I don't really want to know if I have the breast cancer gene or the high cholesterol gene, then you don't need to know this information if you don't want to know it. And that's just a little website for where you can get more information. Like I said, there's also ongoing opportunities. So when you sign up, this is what your invitation letter will look like. If we send you something to participate in another study, again, they're mainly online. So this is kind of what it would look like if we email you. Again, we're not calling you, just emailing you once you've signed up for additional studies. And they might mail you a questionnaire about sleeping and diet or early diagnosis, etc. And then again, we will try to connect you up with other people who have similar issues, because we want you to feel like you're part of a community and that are people who are going through the same thing that you are. And so again, you make the decision if you want to connect up and do these other things. And you're probably wondering, okay, how can I do this? So if you're a parent, we can schedule a Zoom session and we can just walk you through it. You can just sign up yourself on the couch, or Michaela can come to your house if you're in San Diego. And we're going to be getting a coordinator for the Phoenix site, and they can come to your house. And so it's super easy. And you can just contact Michaela at m1gets@health.ucsd.edu. If you're watching this and you are not a parent of a child with autism, but let's say you're a treatment provider or you're a doctor and you want to get the word out to your patients, you can contact me or Michaela, and we can set up a Zoom to go over some details with you, or Chris Smith can. He's the PI of the Phoenix site, and Michaela can as well. We can give you some brochures for your waiting room, or more detailed brochures that you can go over with families when you're seeing them. And that is basically the overview of this project. And I want to thank you for joining us today. Again, this is Michaela, who's a person you can sign up with. But now I'm going to answer a couple of questions. And I'm going to start out with a couple of questions that we always get asked. So one, if a genetic cause is found, will my health insurance coverage be affected? No, under the Federal Genetic Information Non-Discrimination Act, having genes linked to autism will not affect your existing health coverage or whether you qualify for health coverage. So you can go to the GINA website. So it's not going to affect anything. What happens after my saliva sample? What happens to my DNA after it's been sequenced? We will keep your saliva sample in a secure laboratory. If we've received enough DNA, research staff may separate the sample into smaller amounts and freeze them, and they will be available for an indefinite amount of time so we can continue to do more research. But again, it's always stripped of your identifying information. And then another question sometimes we ask is like, oh, does Spark sell my samples to anyone? No, we never ever sell it. It's just for us. We've been doing this now for 7 years and we still have everything securely in a freezer, and it is only for our research use. It's not for anyone else. And now I will look and see a couple of questions. Okay, so here's a question. It says, I have an adopted child with autism diagnosis. Can I still participate? Yes, you totally can participate. Your child will provide the saliva sample, but because you're not genetically related to your child, your sample, though, will not be sequenced, just your child's. But you're still part of the project. You still get the gift card and the webinars and the return results if there's anything, if there's anything relevant for your child. Here's another question. I have 2 children, but only one has a diagnosis of autism. Can I sign up both children? Yes, actually, I forgot to mention that. Thank you so much for raising that. We want the whole family. We want mom, dad, siblings. If you've got 10 children, we want all 10 children to provide a saliva sample, because that helps us really hone in and narrow in on what's going on genetically in autism. Oh, here's another good question. Is there an age range for participants? There is no age range. If you are watching this and you have autism and you're 80 years old, then you can yes, we want you. So in fact, it turns out, if you look at the distribution of the data that we have in the Spark data set, we don't have enough people who are actually older. Our average age is about 8 years old. And as you get past like 30, we don't really have enough. So we really want all ages. So here at our center, we actually can provide a diagnosis of autism as young as 12 months. So we've got kids, as young as like 12 to 24 months, as well as, we want as old as possible. So any age is Perfect. And let's see, I think, oh, this is so nice. This person, Deborah, is being so nice. Thanks, Deborah. She says, do you want us to solicit friends of ours who are out of state? Yes, that would be really wonderful. And they can sign up through our San Diego site, or if their site has, because there are lots of different, lots of different states that are participating, they can, sign up with that. Like Georgia, for example, has a Spark site. If they live in Atlanta, they can sign up through them. But of course, we want everybody to sign up through our site in San Diego. So thank you. I live in Chicago. Can I participate? Yes, anybody in the entire country can participate. Just go to our website. You can totally sign up through UCSD. It doesn't matter. You don't have to live in San Diego. Oh, this is a good question. I had a hard time getting saliva for 23andMe for my child. Can I use the sponges from our kits for the 23andMe kits? I don't really know, because I have never tried that. And plus, you would need the sponges to participate in our project. However, you can email Michaela and she can see if she can get, if she can get it. I mean, the worst that can happen is you could try, right? We can send you the sponge. You can use it for 23andMe and see if you can get enough saliva out. I don't know if if the quantity of saliva they need is the same as ours, but you totally can try it. I'm sure Michaela can mail you some sponges so you can totally try that. Let's see. I have 2 diagnosed children with autism, but they have different fathers. Both fathers are willing to participate. Is that possible? Absolutely. Yes. We can totally, we would love everybody to participate. So that's great. There might be some complications. Michaela will figure out about having maybe 2 separate registrations. I'm not positive, but yes, we definitely want everybody. Well, this is a good question too. You guys are asking really good questions. If it is found that a child and parent have genetic indicators of autism, do you then ask for the grandparents to participate? No, we do not. I think the grandparents can participate. I'm pretty sure that the entire extended family is more than welcome. But let me go back and check on that. We are going to actually publish all of these questions. So like, this is the first one that I'm not 100% sure of the answer. I know we're not going to go and ask the grandparents, but the question is, can they just, can they provide a saliva sample? I think they probably can. But I'm going to double check on that one. Then there's another question. 2 children have autism, except one has Down syndrome as well. Yes, absolutely. We would love to have both children participate. And then here's another question. I have a son with autism and we got genetic testing done with a genetic doctor that we saw before. Can we still participate? Absolutely. And for me as a scientist, I think it's actually a great idea, because it's interesting to see if the genetic finding you got from your doctor's office actually you're going to get the same genetic finding from Spark. So far, that has been the case. If you have a genetic finding somewhere else, you're going to have it with us. The only thing to note, though, is it depends on the rigor of your doctor's offices. Like what we do here is it's got to be, once we find genetic finding, then another, like a CLIA certified lab checks it to make sure it's accurate. And so any finding that we send out, we have literally double and triple checked. So I don't know the rigor of your doctor's office's policies. If they're not as rigorous, then maybe the finding that they gave you may or may not be correct. It's probably correct. And so almost always there's concordance between the genetic findings, but it would be interesting. You can ask your doctor, say, hey, were my genetic results certified by a CLIA lab? What was the verification for this genetic finding? Let's see, another question. My child is diagnosed, but mom and dad aren't, although we suspect that both actually have it. Will the survey results help us get adult diagnoses? Not really. I think that if you really want to get an adult diagnosis, like it's very hard to diagnose autism just based on a survey. You're better off going to your local hospital, finding out if there's a developmental evaluation clinic or somewhere that specializes in adult diagnoses. It's going to be better for you because you'll get better information. That's going to be more accurate. The screening questionnaires are great, but the accuracy is not half as good as when you go in person. Yes, are you going to send the link and all about how to register? Yes, Michaela, why don't you send that link, because if I exit here, it's going to lose it. Yes, that's a really smart thing. The link is actually in the presentation, and we'll email it to you guys. But if Michaela, if you want to put that in the Q&A right now, then families can just copy it from here, right here. But yeah, it's it was on one of the slides. It's like Spark for Autism /UCSD.SARC or something like that. I'm pretty sure that's what it is, but without going back into the slide, I mean, I guess I can quickly see, is it on here? Yeah, here it is. So basically, this is how you sign up: sparkforautism.org/UCSD.SARC. You can take a picture of it with your phone. You can go there right now and you can sign up as soon as this webinar is over. And we have 3 adopted children, 1 with an official autism diagnosis. If we obtain a positive finding for the children without an active diagnosis, does that indicate an official diagnosis? Interesting question. I wouldn't say that it definitely indicates an official diagnosis, but it would suggest that you should go and try to get a professional diagnosis. The reason I say that is because sometimes children who don't have autism will have a similar genetic issue. Most of the time that is not the case, but it's not completely unheard of that could happen. And so it's suggestive of autism, but it's probably worth it to go ahead and get a diagnosis if your allegedly, unaffected child comes back with a positive genetic finding. Somebody says, is this whole exome sequencing or a CMA test? Yes, this is whole exome sequencing. And again, the exome is only around 1% of the human genome, but it's a super critical part because it's the part that encodes for proteins, that creates the instructions for creating us. And so that is what we're doing. Yeah. Occasionally they will do like whole genome sequencing, but that's just huge. And really, we're just more interested in these protein encoding regions. Let's see. All right. This has been really wonderful. We've gone over time, and I've answered almost all the questions. Here's right. I'll do this last one. This is a really good one by Sarah. If we don't receive a genetic finding, then does that mean that my daughter's autism is caused by environmental factors? No. As I said, we still haven't figured out all of the genes related to autism. Right now, we have only found genes related to autism for around 10% of autism cases. The other 90% we don't know, because it's complicated. There's a lot of like background genetics. So if your child has a particular genetic profile in the background and a particular combination of that, even though there are no single genetic, mutations or changes that can be observed, it's this particular background that they have that's related to autism. So if you don't get a finding, it doesn't mean your child's autism was environmentally caused. Maybe one day when we find out and we understand the genetics of autism perfectly, you can make that inference. But right now, that's why we're doing this study. So one day we could get there. So, all right, you guys have been really wonderful. I'm very excited about how engaged you all were, and it was really nice. I wish I could, see you in person, but I really appreciate you tuning in. And until next time, thank you so much. Bye.